So with the last video, we talked a lot about the autorhythmic cells that make up the intrinsic conduction system of the heart. You'll remember that those are the 1% of cells in the heart wall and that they generate electrical signals or action potentials that travel across the heart and tell the heart muscle to contract. You'll also remember that these cells are kind of interspersed throughout the heart muscle wall, and they're represented in areas that you can see in yellow on the diagram here. What we're going to focus on today are the other cells, the 99% of cells that make up the heart muscle. So these are known as non-autorhythmic cells, and as their name implies, they do not have a self-rhythm. So they don't generate action potentials, they don't generate electrical signals. Their role instead is just to contract. So they receive those electrical signals from the autorhythmic cells, and these non-autorhythmic cells are going to contract in response to them. If you look at this picture here, this should be a slide that hopefully looks familiar to you from 227. So this is cardiac muscle tissue, and you may remember from 227 that one of the hallmarks of this type of tissue is the intercalated discs that you can see. So I'm circling a few of these here. You may remember that these are the dark staining bars that are kind of perpendicular to the grain of the tissue. When we look at cardiac muscle cells, they are V-shaped. So I'm going to draw what I mean by that here. We've basically got these branched cells. So they're V-shaped. There's a little nucleus. Sometimes they're Y-shaped. So I'm drawing a little Y-shaped cell in here as well, a little nucleus there. And basically these intercalated discs are the junctions in between two of these non-autorhythmic cells. So any place that we're seeing these dark staining bars, what we're actually seeing is a junction between two of these non-autorhythmic cells. The intercalated discs are important because when these non-autorhythmic cells receive an action potential from the autorhythmic cells, that action potential travels across the cell, and then this intercalated disc or this junction that we have between the cells helps to spread that electrical signal to the adjacent cells. So that's one of the hallmarks, again, of cardiac muscle tissue, and that's why you see these dark staining bars with only cardiac muscle tissue, because they're there to help spread that action potential from one cell to the next. So if you look at these diagrams here, what I want to do for a minute is just focus on the diagram on the left, and what we're seeing is an action potential in a skeletal muscle. So you may remember from 227 that an action potential consists of a depolarization, so I'm kind of running my finger across the depolarization here, followed by a repolarization, and that depolarization plus repolarization is, again, what we refer to as an action potential. And the action potential is the actual signal that tells the skeletal muscle to contract. If you focus for just a minute on the red line that you see right here, this is actually a line representing tension being generated by the muscle. And you'll notice that following that action potential or that electrical signal that tells the muscle to contract, we actually have tension being generated, and we see the muscle contracting in response to that. If you look over here on the right side of our diagram, this is the action potential for cardiac muscle, and you'll notice that it's a little bit different. So ignore for a minute this red line. What I want you to notice is the action potential for cardiac muscle starts out similar to what we see with skeletal muscle. We get this depolarization that I'm running my line or running my pointer across now. And then we start to repolarize, but something stops that, and we go into what's known as the plateau phase before repolarizing again. This plateau phase is really, really important to the function of cardiac muscle, and what I want to do next is talk about why that actually is. So if you look over here and you compare it to this action potential in skeletal muscle, you'll notice that the action potential in skeletal muscle takes just a few milliseconds. So it's done and it's over with really, really quickly. And because of the fact that it's so rapid, we can actually send those action potentials or send those electrical signals to skeletal muscle telling it to contract several times a second. And because we can so rapidly stimulate a skeletal muscle, telling it to contract, basically skeletal muscles don't have enough time in between the signals telling them to contract to actually relax. And this allows skeletal muscles to be able to have a sustained contraction, to kind of hold that contraction for a period of time. If you look over here at cardiac muscle, you'll notice our action potential takes a lot longer because of the plateau phase. So it takes altogether about 250 milliseconds, several hundred times longer, for us to go through the depolarization and repolarization in a cardiac muscle cell. So what that means effectively is only once every quarter of a second are we capable of sending an electrical signal to the heart telling it to contract. So we can't send the signal as quickly, and because we can't send that signal as quickly, the heart muscle is actually not able to have a sustained contraction. There's enough time between in each of these electrical signals that the heart is actually able to relax between contractions. So there's something that's really important about that. If you think about the heart, its role is to act like a pump. And if you're a pump, the contraction phase of a pump is really important because it pushes something out of the pump, right? But the relaxation phase in a pump is just as important as that contraction phase. The reason for this is simply that the relaxation phase allows the pump to be able to fill. So when the heart is relaxing, when it's not contracting, this is the time that it's actually filling with blood so that there's something to pump out, there's something to push with the next contraction. If the heart were able to sustain contraction, it would not be able to function like a pump because it wouldn't be able to fill with blood. And what that means effectively is once it has pumped once and it's pushed the blood out, it can't fill again. That means it can't pump any more blood out, blood flow in the body stops, and basically you would die. So this plateau phase that we see with the electrical signal or with the action potential in cardiac muscle is really, really important to the function of cardiac muscle itself. We need that extension. We need it to take longer before repolarization can occur so that we can't send the signal rapidly to heart muscle to contract, and heart muscle has this kind of obligate pause, this obligate relaxation that allows the heart to be able to fill with blood in between contraction. So what I want to do next, after we've kind of looked at the difference in the action potential between skeletal and cardiac muscle, is talk a little bit about why we see these differences. So we're going to review what's going on with an action potential in skeletal muscle. This is review material from 227, so it's something that you've seen before. And then we're going to compare that to what's actually happening in cardiac muscle and why we see this extension of the repolarization, why it takes longer, why we have this plateau phase with cardiac muscle that isn't seen in skeletal muscle. So if you look at this first slide here, what this is showing us is the action potential of skeletal muscle. And before we get into actually talking about what causes that, what I want to do is just review a little bit about skeletal muscles and what's going on in skeletal muscles when they're at rest, so when they're not actually contracting. So what I have represented in here in pink, kind of this rectangular structure, is a skeletal muscle cell. And one of the things that you may remember about skeletal muscle cells is that they have sodium and they have potassium on the two sides of the membrane. But the sodium is at a higher concentration outside the cell, so I'm using that arrow to represent that, and it's at a lower concentration inside the cell. And potassium is at a higher concentration inside, and it's at a lower concentration outside. And the reason for this has to do with the sodium-potassium pumps. So you may remember that sodium-potassium pumps are always moving sodium out of the cell. And that's the reason that we have this higher concentration of sodium outside of the cell. At the same time that those sodium-potassium pumps are moving sodium out, they are also pumping potassium in. So they're constantly moving potassium inside of the cell, and that's what gives us this higher concentration of potassium inside the cell. There's one other thing about the sodium-potassium pump that you should remember that can help you to understand what's going on in a skeletal muscle cell, and that is that each time the sodium-potassium pump pumps, it's going to move 3 sodium out, and it's going to move 2 potassium in. So what that means is every single time that we go through a contraction of the sodium-potassium pump, we have more positive charge in the form of sodium being moved outside of the cell, and we have less positive charge in the form of potassium being moved inside of the cell. Because our net movement of positive charge is outside of the cell, due to the higher amounts of positively charged sodium moving outside, we end up with a situation where the outside of the cell is positive relative to the inside of the cell. And again, that's caused by the fact that we have more positively charged sodium moving out, and we have less positively charged potassium moving in. You may remember that the difference in charge on the 2 sides of the membrane is equal to negative 70 millivolts, okay? And we refer to that as our resting membrane potential. So if you were to look at this graph here, negative 70 millivolts is about right here. That's our resting membrane potential. And an action potential occurs when the membrane potential, or the difference in charge on the 2 sides of the membrane, changes. So right here is where we've got an action potential developing. And what initially happens to cause an action potential to develop is we get sodium channels opening. And if sodium channels start to open, so I'm going to try and draw some of these sodium channels in. If sodium channels start opening, these are my representations of sodium channels, all across the cell membrane, and we have a higher concentration of sodium outside of the cell than inside, what's going to happen is sodium's going to move down its concentration gradient once these channels have opened, and sodium's going to start to rush inside of the cell. That sets up a situation now where we have more positive charge moving into the cell and less positive charge moving out of the cell. And when we end up in that situation, I'm going to cross out these positive and negatives, we end up actually depolarizing the cell. So we get to a point where the cell is positive on the inside, again, because so much positive charge is now moving into the cell in the form of sodium. And relative to that, we are negative on the outside. And when this happens, again, caused by sodium channels opening, we refer to it as depolarization. And depolarization is what's happening on this initial part of the action potential here. So the next thing that happens, and this is going to happen right here on this line, you'll notice our action potential stops, so we stop depolarizing, and we end up repolarizing, meaning that the line reverses and it starts moving back down. The reason for that repolarization is that the sodium channels close, so that stops us from depolarizing further. Otherwise, we just continue extending that line up indefinitely. But those sodium channels close, and instead we get opening of potassium channels. So I'm going to use green now to represent potassium channels opening. So I'm drawing some potassium channels in. These potassium channels are opening all across the membrane. You guys know that potassium is at a higher concentration inside of the cell. So in that case, what's going to happen is potassium is going to start to move out these channels that have opened, down its concentration gradient from an area of higher concentration inside to an area of lower concentration outside. When that happens, all of a sudden, once again, most of the positive charge is moving in this direction from inside to outside in the form of potassium. And because we now, once again, have more positive charge moving outside than in, we repolarize the cell. So repolarization is what's happening on this second part of the graph right here, when we're going back towards the original membrane potential. And you'll notice that because of this net movement of positive charge, because most of our positive charge is moving out of the cell in the form of potassium, we go back to positive on the outside of the cell and negative on the inside of the cell. And again, that's what's known as repolarization. So we're going back to the original state of the cell, again, where it was positive on the outside and negative on the inside. That depolarization plus repolarization is the electrical signal. It's the action potential that tells the muscle to contract. And you'll notice if you look at this graph that once we've gone through that depolarization followed by a repolarization, that the muscle starts to generate some tension. So that tension is represented by this red line. You'll see tension is increasing. This is representing a muscle contraction that's happening following the electrical signal or following the action potential that told the skeletal muscle actually to contract. So with this last slide, what we're going to do is look at the situation for what's happening with contraction in cardiac muscle. And the contraction part of cardiac muscle is basically identical to what happens with skeletal muscle. There's a little bit of a difference in the action potential that we talked about previously, whereby that action potential is extended. So we have that plateau phase that takes or makes it a lot longer time for repolarization to actually occur following the depolarization. And again, there's a reason for that. It ensures that the heart is able to relax in between contractions so that it can fill with blood and it can continue to push blood out to the tissues of the body. So I mentioned previously that cardiac muscle has basically the same situation going on as skeletal muscle. So due to sodium-potassium pumps in the resting state, so when a cardiac muscle is not contracting, we have more sodium outside and we have less sodium inside. We've got more potassium inside and we've got less potassium outside. And because those sodium-potassium pumps are working in an identical manner as they are in skeletal muscle, we've also got this resting membrane potential, or this difference in charge on the outside versus the inside of the membrane in a cardiac muscle cell, whereby the outside is positive and the inside is negative. The one thing that's different about these cardiac muscle cells that we don't see in skeletal muscle is that we also have calcium channels. In addition to the sodium and potassium channels, we have calcium channels that are embedded in the cell membrane of cardiac muscle cells. And we also have a situation where there's a much higher concentration of calcium outside of a cardiac muscle cell than there is inside of a cardiac muscle cell. So this is kind of our baseline situation, what's happening with cardiac muscle cells that's a little bit different from what we see with skeletal muscle cells. Other than that, what happens to initiate an action potential in cardiac muscle is effectively the same as what we see in skeletal muscle. So our resting membrane potential, or our difference in charge on the two sides of the membrane, is equal to -70, just like it is in skeletal muscle. So I'm drawing that right here. And just like we saw in the previous graph, here is the place, okay, where our action potential is initiated. And just like what we saw with skeletal muscle, what actually causes the action potential to be initiated in cardiac muscle is the opening of sodium channels. So we get all of these sodium channels that are opening across the cell membrane in cardiac muscle, and that causes sodium to start to move through these channels into the cell. So it's just moving from its area of higher concentration outside of the cell into its area of lower concentration, which is inside of the cell, and that causes this depolarization of the cell. However, remember, we also have calcium channels in cardiac muscle, and sodium channels in skeletal muscle and cardiac muscle are also sometimes referred to as fast-gated sodium channels. The reason for that is these sodium channels Once they're told to open, they open very rapidly and they close very rapidly. So they're very fast at opening and closing their gates, and so we refer to them as fast-gated channels. Basically, once they get that signal that tells them to open, they pop open, and very rapidly after that, these channels also close. The calcium channels that are also found in cardiac muscle cells membranes are what are known as slow-gated. So these slow-gated channels actually get the signal to open at the same time as the sodium channels. The sodium channels, upon getting that signal, just pop open and they start letting sodium rush in. The calcium channels are much slower. So once they receive that signal to open up, they start to very, very, very slowly open. And actually what we see happening is that the sodium channels close, and then we get potassium channels opening. So once again, I'm going to use green to represent potassium channels opening all across the membrane. And you guys know potassium's at a higher concentration inside the cell. So potassium starts to move out of the cell, and now we have our net movement of positive charge out of the cell. And you'll notice after those sodium channels close right here, that initially we start to dip back down, but instead of repolarizing immediately, okay, which this green line would represent an immediate repolarization, instead of that happening, like what happens with skeletal muscle cells, instead we go into kind of this plateau phase. And the reason for that is right here, okay? The calcium channels finally respond to that signal to open. So I'm going to use, I think, purple to represent calcium channels. So we get this situation where potassium's moving out, and finally those slow-gated calcium channels are opening all across the cell membrane. And we've talked about this idea that calcium's at a higher concentration outside. So when calcium channels open, calcium, which is a positive charge, starts to move inside of the cell. So we have potassium moving out, and normally potassium moving out would immediately repolarize the cell. But we finally get those calcium channels open, and now each potassium that's moving out is basically being replaced by a calcium that's moving in, and so we're no longer repolarizing, we're no longer depolarizing, we're just kind of in this situation where we're really just kind of revving our wheels, and our membrane potential isn't really changing for an extended amount of time. So what then happens is our calcium channels close. So they close, however, when they close, those potassium channels are still open. So I'm going to redraw those potassium channels back in. They're still open, and they're still allowing that potassium to leak out of the cell. So right about here, okay, is where those calcium channels close. So calcium is no longer entering. Now we just have potassium exiting, and that's going to repolarize our cell finally. And that's what extends that plateau phase, is the opening of calcium channels, basically acting against the potassium channels that are opening, with calcium entering, counteracting the effects of potassium exiting, and now we put this plateau phase in there that extends the action potential and allows us to be able to have those obligate relaxations in between contractions in cardiac muscle.